what do you call the other one, Zoom. So also I'm a little unwell, but let's to make it nice. What would be nice is that if we engage with each other, I think it's more like a class than like a presentation, and so therefore please keep on asking me. So interrupt me whenever you want to ask something. So it can be, I guess, Dr. Khan, it's okay, right?
Yeah,
That,
yeah, fine, fine. Yeah.
okay. So keep on asking me questions as and when it occurs to you, and I will also keep on engaging with you. Probably I'll be asking you questions. So what I am going to give a total of 3 classes or talks today, tomorrow, and day after, and in these I thought I'll present to you an overview of the subject of cytometry, the trends, what cytometry is about, first of all, going by that, what has been the trends in it, and where is it leading to. So just basically give you an overall. It won't be a very technical, what we call as a very pedagogic class, okay, but basically presenting, trying to present ideas. So the first thing, the first one that I plan to give is on what I call as single cell analysis and flow cytometry. This is okay. This is the difference of being actually in the physical class and online class. How many? I mean, I guess how many of you have heard about flow cytometry? I guess most of have you heard of flow cytometry?
Yes, sir.
Yes, yes, yes.
Okay. And anybody heard of single cell analysis?
Yes, sir.
Okay. So do you feel there is? I mean, why do you feel is there? I mean, is there a connection between single cell analysis and flow cytometry?
So flow cytometry is used for, cell counting, whenever you see the technique, if you see that
Yeah.
in a solution, like each cell is passed through the laser beam, and that's how the detector, counts
Exactly.
the. So
So
that.
that's the key word. Each cell. Okay. So the measurement happens in at one cell at a time, and although flow cytometry started off way back in history, several decades back, but then this idea of trying to look at one cell at a time has come only in the last around 15 years or so, more so. So we'll first begin with we'll actually dealing with why should one look at one cell at a time? What is the basis? So the first thing is that first of all, one has to look at us whenever one actually looks at bodily function. One has to look at the basic unit of life is one cell. It's not the DNA molecule. It's not a gene. It's not the nucleus, but it's the cell. Because only at the level of the cell do we see life. We don't see life at the level of the DNA or the proteins, right? Now, the other thing is that whenever, even if you have, let's say, uh, a clonal population of cells. So I think the word clonal is familiar with you, right? So,
Yes.
so whenever you see a clonal population of cells, and this you actually see frequently when you grow cells in culture. In cells in culture, what happens is you grow from one single cell, you can actually grow a full plate, a full flask of cells. So all these cells, they can be assumed to be clonal because they started from one. And by clonal, we mean that they genetically are identical, although they are genetically identical. Now, such clones we keep on generating when we do transfections. That means we are trying to introduce a gene into a cell, and in many situations, the genes that we introduce, it could be linked to a fluorescent protein. It could be a gene of fluorescent protein. And because it is a clonal population, all cells are genetically identical. We feel that the control of expression of this fluorescent protein, let's say GFP, would be the same in all cells. But still, if we look at a clonal population of cells which have, in which you began with a single cell, you transfected a single cell with a fluorescent protein, and you gained a clonal population, you find differences in the population of in the expression of GFP. And how do you look at it? If let's say you have a flask of cells, and if you look under a fluorescence microscope, and you start seeing that by the green color here, you start seeing that some cells have. Let's see if my laser pointer works. No, probably it only works in the full screen mode. Okay, so are you being able to see the microscope, the cursor? I don't know whether you're able to see.
Yes.
Okay, so here you see that although most cells are expressing by the amount of light that you see the same level of green color intensity, but you see some cells which are expressing more, although they are all genetically identical. And these cells that you see them expressing more are few in number. So, and these differences, so one thing goes on to say that is that although these cells are genetically identical, but still you see differences in the expression of the fluorescent protein. And these could be because not of genetic changes at the level of DNA, but changes at the level of, let's say, RNA and protein, which control how much of the gene will be transcribed and therefore how much of protein will be made. And these and probably these controls are because it has local environments are different. Let's say the local environment is different. There is more of cell crowding, and that makes, let's say, some cells in this region to express more of this protein. Or it could be that these cells here, they have a nutrient condition which is in this small locality here, which is different from the overall nutrient condition there. There could be small changes in small differences in concentration of nutrients, which can actually decide these. So therefore, if we had, so we see so the important message is that although we are seeing differences, although most cells are expressing the same, but still there are only a few cells which are different. And let's say if we were to take the total, if we were to look at the expression of the GFP from all these cells together, we would have got one value, and that would have therefore missed these differences that some cells are expressing more and some less. So this is in culture. When we go inside the body, we also find we find something similar. We find that in the body, for example, when cells are differentiating, we find that when differentiation happens either in the skin or in the blood, that some cells which are giving rise to the different differentiated cells, which can be called as founder cells or stem cells, they are producing the entire cells of the differentiated population. And these cells are generally, they're very few in number. And so therefore, if you want to act, and if you want to find out to detect these stem cells which are few in number, you will miss them if you actually, let's say, look at the activity of all the differentiated cells together. So only if you analyze, if you look at if you look at the entire differentiated population, and if you look at cells, one cell at a time, you realize that there are a very few cells. Let's say if you had 1,000 cells, there are around 5 cells in the total population which are different, and probably they are stem cells. And but as the stem cells differentiate, their numbers also multiply and they become more, and therefore you will find that there are more in number of differentiated cells which you actually pick up. So this, you would not have been able to detect these stem cells if you are looking at the entire population of differentiated cells. So this is again conveying the same picture, is that, let's say, if we have some cells which are growing in time as they're multiplying in time, and as they multiply, you find that you have one level of protein. There's a particular protein present in all the cells which are present at a particular quantity, and this quantity is reflected in the peak that you see here. But now, as time goes, you find that a certain population of cells, they start accumulating more protein than the others, which are lighter in color, and these ones are dark in color. But you are looking at the total protein by, let's say, a Western blot, and what you see is, when you're looking at the total population of cells, you are being able to only pick up one value of the protein amount, which is the averaged value present in the entire population of cells. You are not being able to pick up these differences, that there are two populations of cells, one having more protein and the other having less protein. And this could have been only possible that if you were looking at one cell at a time, and then you would have found, when you were looking at one cell at a time, you would have found that there are actually two populations of cells which have two different protein amounts. A similar story being repeated here in this slide is that we see here on the left-hand side panel a population of 5 differentiated cells, and we are looking at this mRNA transcript which is there, and we find that if we look at the distribution of this transcript in all these 5 cells together, we just get one value. We won't be able to pick up that there is one cell which has more of this, one cell which has less, and the other cells which have none. So this would have been possible if you had looked, therefore, at each individual cell, that what is the level of transcript in these 5 cells, and would have found that yes, in these 4 cells, the level of transcript is 0. Here there is 1 molecule of the transcript, and here there are 5. Only when you have looked at 5. And then when we would have done, you would have looked at one cell at a time, you would have found that, therefore, that there are 3 populations of cells. A population of cells larger in number, which is, a population of cells which is larger in number, which has 0. That's one population. Another population which is more than that, which has 1 transcript, and another one which has only 1 cell and that has 5 transcripts. So we find that there are actually 3 populations of cells. We can actually call these as subpopulations in this total population. And it could be that one cell which has more, the number of transcripts being more, it is probably an important effector cells, because it is probably driving the population because of its higher level of mRNA molecules of a particular type into doing something different, and therefore that makes it different. And this is what exactly happened. So this is the problem of looking at of a total population of cells together. Now, when it came to tumor therapy, as you see on the right hand side of the slide, now in tumors there is a concept that there is a single stem cell which is from which are produced the entire tumor cell mass. And this stem cell is, as we said, is actually few in number. They are few in number. Now, the initial strategies to kill the tumor were devised on trying to look at marker proteins on the entire population of cells. And so when a therapy was decided based on killing based on a protein which is present on the entire population, that means when the decoction was made from this entire tumor and a protein was detected which was found which could be used to actually to make a drug which will kill the entire tumor, when this drug was given to the tumor, what happened was all the tumor cells died. But because this drug was designed against which was present in all the tumor cells, in the tumor cells, it did not kill the stem cell. The stem cell survived, and after some time of giving the drug, there was the tumor cells grew back because the stem cell was alive, and so there was a relapse of the tumor. So if you now actually devise a strategy by looking at by identifying a protein which is present only in the stem cell and not in the tumor cells, then you can devise a drug which will only kill the stem cell and it can avoid the other cells. It doesn't matter. And so then it was found that when such a drug molecule is made, made and given to the entire tumor mass, then the stem cell was killed, and therefore there was no relapse of the tumor because the stem cell which was producing all the other tumor cells was not there. So the idea is that always in the population of cells, there are a few cells which are different, whose molecular character is different, and which drives the entire population, although they're few in number, these few cells, and because of the different molecular character, they drive the entire population into a particular direction. This is an example from a paper in which it was found that whenever you have cells growing in culture, and if they're multiplying, after they exit after mitosis, most of the population goes into entering the cell cycle. After mitosis, they enter into G1, then they come to S and G2, and they go through the cell cycle. But there is which you actually see as this one, they actually go to the enter the cell cycle. But a small population of cells, they don't enter the cell cycle. They go into the quiescent stage. And it was found that in these cells, there is a particular protein which drives them into the cell cycle. The level of that protein the activity of that protein is very much low, and therefore it's not sufficient to drive them to re-enter the cell cycle. Whereas in the others, the threshold level of that protein is high, and therefore it drives them to go through the entire cell cycle. So therefore, you so therefore this is again this is the, the summary slide of all this put together. You have a population of cells in which there is a mixed population. If you look at the mixed population and you do a bulk analysis, you find that if you are looking at one particular and if you find that if you're doing a bulk analysis for looking the level of a particular protein, you find that most of the cells have the most of the cells are of one population only, and which has the major level of that particular protein. You don't pick up, when you do a bulk analysis of all this put together, you don't pick up other cells which have different levels of the protein. But then if you now do an analysis cell by cell, if you look at one cell at a time, then you find that there are subpopulations. So the entire subject of so this is what cytometry is about. Cytometry is trying to look at all the properties of a cytometry is basically about cytos meaning cell and metry meaning measurement, measuring the properties of a cell, is to look at the properties of a cell and to look at one cell at a time. And here you will measure all the properties of a cell that you can think of. These could be physical properties like the size of a cell. It could be even actually optical properties of a cell. It could be what chemicals are, small molecule chemicals are present inside, metabolic chemicals, the amount of proteins. So this is what cytometry is about. And cytomics is that if you are looking at one cell at a time, and if you're looking at a population of cells, are you being able to find how the activity of individual cells is deciding the behavior of the entire population? That's what cytomics is about. And you will find a population of cells either in culture or in the natural situation in the body. So this is more like a systems approach. Is that if there is a population of cells in culture or inside the body, how the behavior of individual cells is controlling the behavior of the total population. So and another thing is it's being called cytomics. It's an omic, and you would have heard of genomics and proteomics. And what do we mean by genomics there in the word genomics? By omics, we mean that we are looking at all the genes in the and therefore the word genomics in the entire genome. And that's why the omics. So is the case with proteomics. When you say proteomics, you're looking at the all the proteins, and that's why the omics. So cytomics is basically to look at all the cells, all the cells in a particular system, in a particular tissue, organ, and that's all cytomics is about. But omics is different from genomics and proteomics, because in genomics and proteomics, you don't define the unit in which you are looking at these. You just take up a total mass of tissue and find out what all genes are present there or what all proteins are present there. But in cytomics, you say that you decide what all genes are present in a particular cell, or what all proteins are present in a particular cell, and how the protein levels and how the total protein profile is changing from one cell to another.
They're in a single cell.
Yeah, yes. But in a single cell, in a single cell, uh, in a single cell, and you look at this on all the population of cells. I mean, sorry, all the cells in the population, one cell at a time. So let's say you have, let's say you have a piece of liver. So in the piece of liver, you find out the protein, the total protein profile of one cell, and like that you look at the protein profile of each individual cell, and you might land up in finding that although, let's say, in the liver, the most of the chunk is made up of liver parenchymal cells, you might find that depending on which part of the liver you're looking at, you find that a certain population of parenchymal cells, they have a different amount, they have a set of proteins which are different when as compared to cells coming from a different region of the liver. I think I have not made it actually very clear, but
No, it's clear actually.
okay. So, so that is what cytomics is about. That you are trying to you're trying to see everything on a per cell basis, and therefore now can you actually find differences amongst them? Now this concept was vaguely known way back in history, and therefore people thought that one should study one cell at a time. The earliest it began was actually trying to look at, therefore, cells under a microscope, and it began in the 1930s. So the whole story actually began that if you could look at one cell at a time, and because you were looking at one cell at a time, it was difficult to it took a long time, that if you, let's say, if you had a slide of cells and you had to look at one cell at a time, it took a long time to look at all the cells on the slide. So it was very time consuming to look at one cell at a time. It was a very slow and cumbersome process. It started off by this gentleman, Kasperson. He worked on it. He was in the 1930s. He was looking at cancer cells. Actually, he found out when in the 1930s he was looking at cancer cells. He found that the nucleic acid content was different in the cancer cells, and he in the 1930s felt that there must be something like nucleic acids which decide which actually decide that the cancer cells are genetically different from normal cells. But that's how the subject of looking at one cell at a time started. But it was a microscope-based system, and it was time-consuming. Now, later, in somewhere around in the 1950s, there was a project which happened in a defense project which happened in USA, in which they wanted to study a defense project. So they wanted to study a project of biological warfare, where they wanted to study bacteria. And it required that in that project, it was required that these bacteria are studied one cell at a time, because they would be, because these, if there is a biological warfare bacteria which has been introduced into the air, they would be very few in number, and you would have to, if you actually detect the entire sample of air together, you will miss these cells. So you needed to look at one cell at a time, and therefore they needed a system which was fast enough to look at one cell at a time. And they found out that the prevailing method, which was there, of looking at one cell under a microscope was too slow. It would have taken a long time, and you would also have to actually look at a large number of cells, bacterial cells, in order to pick up these few these few cells which are to be used as biological warfare cells. Uh, and so therefore they came upon this device where they said, okay, they came upon this device to actually pass the air through a tube and through a detection system. So they passed air through a tube and detection system, and then they could detect one cell at a time very fast. And from that method came the birth of the method of flow cytometry. Now in flow cytometry, what you do is you have a population of cells in which you have labeled these population of cells to with different antibodies, which are actually looking at different proteins, and then you stain them, and then you pass them and then you pass them through a system wherein you cell one cell at a time, and these cells, they go past. So you can see these cells they're going past one cell at a time, and they go past laser light. And when they go past laser light, then because these are fluorescent, they emit fluorescent light, which is detected. So basically, flow cytometry is therefore derives the name because it's in flow through cells, and so that's why it's called flow cyto, because you can look at cells, and measurement because you are now making measurements of the levels of different proteins present in each cell as they go past this laser light. And when they go past the laser light, they're all fluorescing. And let's say you have cells in which you have labeled different proteins with different antibodies, and each of these antibodies is labeled with a different fluorescent dye. You land up seeing different colors coming out, and from that you can make out the levels of different proteins. So this is what is basically flow cytometry about. And then now when you actually, let me skip this slide. If I come to the next slide, a little better. So when you actually pass a cell through laser light, then one of the three things happen. One is that a portion of the laser light, it gets scattered because every cell has a size, and there is a scattering of light because it hits the light on its surface. A small portion, and that's called forward scattered light. A small portion of the laser light, it enters the cell, and inside the cell there are small particles which again scatter the light, and this scattered light is again picked up by another detector, and that's called a side scatter. And then the cell has been labeled with fluorescent dyes. There are different proteins. Let's say the cell has in the cell, you're looking at 5 different proteins for which you have antibodies, fluorescent antibodies, which are binding to these 5 proteins, and 5 different colors are emitted and they are picked up. So that's what happens. That you pick up a part of the light which is scattered because each cell has a particular size, and that's called forward scatter. And then some of the laser light is getting scattered by the when it enters the cell because of the particulate nature of the cytoplasm, which is there, and then and then while it is getting inside, it is also hitting proteins which have fluorescent antibodies bound to them, and they're emitting light, and that is getting detected. So therefore you actually therefore detect three kinds of light when each cell goes past the laser beam. And here you can see that you see that happening. The laser light is coming, and this is here you see the particles flowing past. When the laser light hits the particle, the ones which are scattered directly, and that's called forward scatter, based on the size of the cells, that's called the forward scattered light. And then the fluorescent light, it goes in a direction 90 degrees to the laser, and it travels to, so now you see these different colors, which means now light of different fluorescent dyes having different colors is coming in this path, along with this side scattered light. The side scattered light is detected in the first detector here, and then different filters, they actually split the light, partition the light into different detectors. And so on the first detector, you see the shorter wavelength green light being detected, and then as you go along, the longer wavelengths, they get detected in this direction. So you have the green, and then the yellow, and then the red. So that's how basically a flow cytometry works. Now, so now if you have, and if you look at what happens in more detail about when one particle is hit by a laser beam, you see that first of all, as this slide is showing you, this is the forward scattered light. You see that the forward scattered light, depending on the size of the cell, the forward scattered light is actually less or more. And depending on how granular is the cytoplasm, the more the granular, the more the light is scattered internally. Okay, and therefore the higher the higher the granularity, the more is the amount of light scattered inside the cell. If it doesn't have these what do you call these particles, the scatter would have been very less, and all the scattered light is also detected. So this is what you. So now, if you just, let's say, in a flow cytometer, what you frequently is done is that you make a preparation of these the blood. Let's say if you look at blood, flow cytometry is used frequently to look at blood. So when you look at blood, if you plot forward scatter based on this axis and side scatter on this axis, and if you see and forward scatter is a measure of cell size, and side scatter is a measure of granularity. On this basis, just on this basis, you are being able to pick up 3 or 4 populations of cells. And you can see here that here you make up this. The smallest are the RBCs. The RBCs have a size of around 5 microns. The next bigger are lymphocytes, which are around 7 microns in size. So you can see forward RBC5 has a forward scatter value which is low, and then you have lymphocytes which have a size of around 7 microns, which have a bigger 7 to 8 or 9 microns, which have more forward scatter. And then you see monocytes. Monocytes are the undifferentiated macrophages. Monocytes, basically macrophages. They have a size of around 15 microns. So the scatter is forward scatter is more. But we also find that, that these cells, they have the lymphocytes as have they are less granular than the monocytes, and the monocytes inside there are more of granular particle available, and therefore you find that because the granularity has gone up, the side scattered light is more, and therefore you find that this value of side scatter is more as compared to the side scatter value for the lymphocytes. If you go up here, you find that the granulocytes, their name is by itself means that they're very granular. They have a very granular cytoplasm. Neutrophils, eosinophils, basophils. Because if you stain them, and if you look under the microscope, you'll find that there are lots of small, speck-like particles inside them. So they have a very granular population, and so therefore they have the maximum side scatter. And now, if you look at, and their size is also more than that of the monocytes. The overall size, they're bigger. These are 15 microns, and granulocytes are around 20 to 30 microns in size. So just based on size, we are being able to differentiate subpopulations of cells inside in the blood. Now, the very same particles, they also are fluorescent, and the way you actually look at fluorescence inside the cell is that what you have to do is that you will have to the way you plot the fluorescence is always there is some unstained cells will also have a background fluorescence, and that is plotted, and that you will have a particular peak which is of low signal, which is having a lower signal height, and also the intensity is also low, the number of events. But after staining, when stain them, then you see the signal above the entire above the unstained cells, and therefore you see an increase in peak height. And then then you start seeing them. Also, you start actually you can plot also the fluorescence also on a 2-parameter plot, wherein you see that this is plotted with one fluorescent dye called phycoerythrin, and this is with another fluorescent dye called FITC. And if you divide into 4 quadrants, you find that there is a population of cells which is low in signal strength, both for P and FITC. These are background cells which have very low fluorescence, and these are the cells which are positive, which have bound a lot of FITC and a lot of phycoerythrin. So there are 2 different populations of cells. So the P was labeled. The P was labeled to one antibody which bound to a population of cells, and to different population of cells as compared to the FITC. So fluorescent, now these data in flow cytometry are plotted in different ways. Usually they are plotted as dots or as histograms. In the dot, each dot represents one cell. And so therefore you have a population of cells here. Each dot is one cell, and therefore all the cells that you've seen here are all the cells that you've passed through the system, to the machine. Let's say if you have passed 1 million cells, all the 1 million cells appear here. And if you look at one dot, that represents one cell, and you get to know what is its corresponding side scatter value and what is its corresponding forward scatter value. And that will be for one particular cell. You can similarly plot a histogram where you build up, where you actually, when you build up a histogram, you get to know that now I see that I have two populations of cells, one with the forward scatter and side scatter value of this much, and another with a higher forward scatter and side scatter value. Also, that whenever we actually look at fluorescence, we have to be, there's always a background fluorescence which is there in cells. And this background fluorescence is because there are always metabolites which are present in these cells, which have their own intrinsic fluorescence. And so this can be a problem, because what we have to when we are detecting specific fluorescence, when we're labeling with antibody, this fluorescence has to be above the background fluorescence, and this background fluorescence is present across a wide range of wavelengths. So therefore, it is important that to know that what is the background fluorescence in these, and so therefore your antibody staining should be sufficiently good enough to be seen above this background fluorescence. Now, because in flow cytometry, you are detecting the different things in a cell by using different fluorescent dyes. There is a wide diversity of fluorescent dyes which is present. And first of all, these dyes are present for, I'm sorry, these fluorescent dyes are present across different wavelengths, right from short UV wavelengths, blue, right through all the visible to the red and the far red. They're present across all wavelengths. And even when you look at, let's say, fluorescent dyes which will bind to DNA, even within DNA, we find a lot of diversity. We find that even just to bind to DNA, we find that there are some dyes which actually emit blue light, some green, and even some red light. So there is a diversity there. And also, I don't know if we're able to see, these dyes also, they have chemical specificity. Some dyes bind to AT rich regions, some bind to GC rich regions of the DNA. And then if you look at, now all of these that I'm showing you are DNA binding dyes. Of these, some dyes they can actually stain, they can stain a live cell. So it is important to know that let's say if you have given a fluorescent dye to a cell, because of a cell's lipid bilayer, charged molecules cannot diffuse through the lipid bilayer. And therefore, so if you want to stain, let's say, cells which are live, in which the lipid membrane is intact, and therefore the charge its outer surface, they won't be able, it's not easy to stain them. Therefore, you need molecules which can actually cross the lipid bilayer. So there are, of these DNA dyes, you have some which actually can go through the lipid bilayer and therefore stain live cells, and some which are only stain dead cells. And then you have a dye here called DRAQ5, which is very live cell, who's which are very live cell friendly. They go inside live cells very easily, and they also emit in the red region. This is important because for live cells, because when you compare let's say DAPI with DRAQ5, to excite DAPI, you will have to use UV wavelengths which are toxic to live cells, and with DRAQ5 you excite it with red wavelengths which are actually very safe. The longer the wavelength, the safer it is. So basically, going on to say that there is a wide variety of spectral ranges for different dyes, a wide variety. Even just within DNA binding dyes, there is a huge amount of diversity of whether of wavelengths that they can produce from blue to red, of chemical regions that they bind. Some bind to only AT and some bind to only GC-rich regions. And then there are some cells, some dyes which are more live cell friendly, being excited by safer wavelengths like DRAQ5 as compared to DAPI, which need to be excited by UV light. So there's a lot of diversity. And these fluorescent dyes are used to detect anything, any activity that is you want to see inside the cell. This is a very well established field wherein you want to detect different proteins are different molecules of RNA and DNA. There is, if you want to look at DNA synthesis, there are different dyes. For, let's say, if you want to look at pH, there are different dyes. For different ions, there are different dyes. So there's anything you want to detect inside cells, there is a dye present. Sorry, this is moving in this. I guess I've put you all to sleep.
Sorry, sir.
No. I thought I have put you all to sleep almost.
No,
okay.
Sir.
No. It's not.
No, sir.
Now, when you have to choose, dyes, what? So now, let's say you're having population of cells in which you want to look at different proteins. Let's say you want to look at different things. So therefore, you are using different fluorescent dyes. Okay. But one thing to remember is that there's an issue with fluorescence. Whenever you look at the fluorescence of any particular dye, you find that it is present across a range. The emission of the fluorescence is present across a range of wavelengths. And if I actually go back to one of the previous slides, in this figure, when you see this particular detector, for example, it is detecting, this particular detector is detecting green light. How is just green light passing through? Because in front of the detector, there is a filter. And in that filter, it not only decides, just allows green, you can say which wavelength range in the green do you want to pass through the detector. Similarly, in this detector, you have another detector in which you can decide which range of wavelengths first it has to of the yellow you want to pass. So there's a filter which decides. Now, if I come back to the slide that I was showing. Now, I told you that each dye has an emission profile, and the emission profile is wide. it is the width of the profile is what is the problem. Because now, if I choose another dye, I find that for another dye to look at a different thing, I find that these two profiles are overlapping. And what I'm finding is that a portion of, and this is you, I find that there is overlapping. And let's say if I now had a filter in front of this to detect this particular, if this light was going to one particular detector, and if in front of that detector I have a particular filter to only detect light of this wavelength range, I will have to avoid light in this overlapping wavelength range, because the filter, because in this filter, the light from this dye will also creep through, because there's an overlap. So what will happen is, in my detector for this, I will detect light coming from this dye, and also light coming from this dye. Now this can pose a problem. I would like to collect only light from this. So one way out is that I collect light only from a region. You see here, I don't know whether you can see it on your screen, where there is no overlap happening. But the problem is, in this region, the peak of the fluorescence is here, and I am at this level. The peak of the intensity of the second dye is very, very low. So it can actually become difficult to detect, to be able to pick up only to use this dye in combination with this, because most of the profile of the second dye, which has a lot, where the intensity is good, it is overlapping with this. Therefore, one solution could be that I say that, okay, that can I, along with this dye, take a dye which is this one, which is far away from it, and therefore there is no overlap. You see here, and you see here, there is no overlap. So then I can actually select, therefore, the green and the red. I can red, and I can detect it without without having any of that overlap problem. This overlap problem is called crosstalk. But in a flow cytometer, in a flow cytometer, the issue is that, first of all, you have to decide pairs of dyes which you choose to detect, which should not be overlapping, and this therefore limits the number of dyes that you can use together. It becomes a problem, because as you keep on, increasing the number of dyes, the whole space becomes crowded, and it will be very difficult to see that you have a pair of dyes which don't overlap, and therefore you manage to collect pure signal and don't collect signal from the overlap region, because the overlap signal will always have light coming from another dye too. So this is one issue with flow cytometry, that you cannot, because the white profiles, you cannot you cannot actually use it to detect a large number of fluorescent dyes, and therefore you can't look at a large number of molecules inside, let's say, a cell. But anyway, if you have an overlap, you will have to remove the overlap, and that is done by a approach called compensation. And this is what this slide is: compensation. is telling you about, that you determine that how much of the signal from the first dye is creeping into the detector of the second dye, and digitally subtract that light from the signal coming to the second dye. There's a way to do it, and that digital subtraction of the contaminating light is called as compensation. So, but what happens is that when you actually digitally subtract light, then what happens is from the overlap region in this you lose light, and as you see here in this particular case, you are losing when you subtract light from the overlap region, you actually lose light even from light which belongs to this particular fluorescent profile, and therefore the intensity reduces, and the intensity reduces, which means if you had a bright dye, even after compensation, you would have managed to be able to detect sufficient signal that would have been still residual after subtraction for this. But if it was a weak dye, then the entire signal can almost totally disappear. Very little signal might be left, which is actually not contaminating, and therefore weak dyes won't work. You need so therefore you need bright dyes to work. So again, this limits that you also, when you're using multiple dyes, you need to use all bright dyes, so that even after compensation there is no signal loss. There is sufficient signal left to detect. So how many fluorochromes can one use in a flow cytometer? Usually people detect up to 1 to 3. Generally is comfortable. People do 6 to 8, but then people, when they want to look at actually more things inside cells, and they will actually need to use more fluorochromes, and the number of dyes goes up. But as the number of dyes goes up, the complications of overlap come in, and it becomes difficult to actually keep on detecting what you call large. To keep on detecting signals when you're using a large number of fluorescent dyes. So this therefore also limits how many protein molecules, for example, you can see, which are labeled with antibodies, which are labeled with dyes. Even ultimately, how many of them can you see together? That limits that ability. Now, we are being able to detect in a flow cytometer. One is being able to detect single cells. Can one now sort cells? Because can you, let's say if you have two populations of cells, one having a particular protein, another not having a particular protein, and being labeled by two different dyes, can you sort them out? Yes, with the flow cytometer comes a mechanism called the fluorescence activated cell sorter, in which dyes having a particular dye wavelength can be given a charge, and these are given a positive charge. So now when so let's say you have a combination of two dyes. Cells you have a population of two kinds of cells, one which are labeled with a green dye and the other with a red dye. Cells only having the green dye, they are given a charge, And so after they exit this nozzle that you see here, they, because of the positive charge, there are two different charged plates that you see here. The cells having the positive charge, they go towards, in this case, it has been reversed. It has been given a negative charge, and the cells having a negative charge are no, I'm sorry, right. The cells having a positive charge, they go towards the negative plate, and they get collected here. So cells having one particular charge, so in this case, cells having a positive charge, they go towards the negatively charged plate, and they're collected here. And all the other cells, they go to another bucket, which are not charged, and they're collected here. So you can actually sort cells out based on their wavelengths, and which actually, and the wavelength decides what antibody that they're bound to, and therefore the antibody decides what protein they're bound to. So cells having a particular level of cells having a particular kind of protein can go to one tube, and the other cells which don't have the particular protein go to the other tube. But now that we can actually sort cells, can we now collect cells in a way that you can collect one cell in one particular bucket alone? And that's possible. Is that, let's say you must have seen a 96 well plate. So a 96 well plate is a plate in which there are 96 wells. Now you can place this well plate under the tube from which cells come, and the plate can be moved in such a way that one cell gets collected in each well. Only one cell goes to one well, and therefore you have 96 different kinds of cells collected in a full plate. Now, we began the story today by saying that we would like to do single cell analysis. Here we did do single cell analysis at this level in a flow cytometer by looking at by using fluorescent dyes and looking at different cell types at a particular level. Now, after, let's say, you have sorted these cells out, would you like to look at other differences amongst these cells? Other differences can mean, would you like to look at now the total protein profile in these cells on a per cell basis? Or would you like to look at the total number of RNA molecules, and that is the transcriptome in these cells? That is therefore possible that now because you have in this well plate, you have actually in each well plate you have one cell there. You can now do a total protein analysis by mass spectroscopy, and now look at the total proteome of each cell on a per cell basis. And for 96 cells, 6 cells, you can do the proteome analysis. And similarly, you can, if you want to do, you can look at by sequencing, by using a next gen sequencing machine, you can look at all the RNA molecules on a per cell basis. So this is what therefore brings us to the subject of single cell multi-omics. That on a single cell you can look at anything. You can do total omic analysis. You can look at the total protein amount. You can look at the total number of RNA molecules. You can look at the total number, the total metabolite profile or the lipid profile on a per cell basis. That's the end of my this. I think this was it was presented in a in not at all a good way. But anyway, if you have any questions, I am.
So, excuse me, sir. Excuse
Yeah,
me, sir.
yeah, yeah. Yes, please.
So first of all, it was a very amazing lecture. I learned a
Uh,
lot through it. And
I mean, I feel you probably. I hope you're serious because I actually got lost while doing this. Anyway, yeah, you asked whatever you asked. Yeah.
no, sir, it was very amazing for me. It was very informative. Sir, my question is that you said, like, you were discussing about cancer cells.
Mm-hmm.
In cancer cells, I want to know why leukemia is a major problem. Like, why drugs are not being able to develop against leukemia. Like, is there not a stem cell that is, originating all these tumor cells, or like we are not able to find the protein marker in the stem cell?
I guess it must be that they're not being able to find the protein markers, probably. But I'm not a. I mean, I actually come from a from. I'm not a. I'm don't basically come from a biology background. I mean, in that sense. But I think here it must be the problem that not being able to find the stem cell basically.
In fact, I feel that with this single cell proteomics and genomics, I think today, not very far off, we'll be able to find out what is really the protein being expressed in that particular cancerous cell. So it's only a matter of few years probably when you'll have a definitive marker, just like earlier markers were developed. So Ayushi, my this thing is that is where what you have learning single cell proteomics and genomics will suddenly open up an avenue which you never knew. Like what is the transcriptome being produced by next generation? You can look at it, or you can look at the protein population, and from that say, okay, if this protein is present, because now we can sequence every protein in a single cell with the new method, because we are able to even count as much as 600 proteins in a cell. If it is present, it can measure what is called sequencing. So I think it is just a matter of time before the method is standard. So what you don't see today is because it is not standardized yet, that it is reproducible. So that will take another few years. Methods will be standardized, and soon what the question you have raised will be answered, I'm pretty sure. Is it satisfactory, Ayushi?
Yes, sir. Thank you so much, sir.
Yeah. So that's why you must learn this workshop. You will learn a lot of new things happening for tomorrow, which is more important for you. There is nothing called not possible. With technology advancing, many new things are going to be possible, and that is the optimism you should have. Am I right, Amit?
Yes, I mean, I mean, more than teach you about, flow cytometry, it basically will tell you that the single cell approach. Why is it important? Okay, this is and this is a thing which is happening, and also why is flow cytometry a single cell approach? Why is it a single cell method? Basically, will tell you that.
Is there any other questions, children?
Yes, sir. I have a question.
Yeah, Anushka asked.
So you said that.
Mm.
Yes, one of the limitation which contributes to background noise is autofluorescence, right? So how it is overcome then?
Yeah. Okay. So a couple of ways. One way is that there are, before you stain, you can always use a chemical method that will bleach the autofluorescence in a tissue. That's a method. That's one method. The other methods is that how to tell you, because
No.
The problem with autofluorescence is it is present across a wide range of wavelengths. Okay, and So now there is an approach which is used, which is called as spectral unmixing. And what is that? Is that you say that you know what is the spectral profile of each of your fluorescent probes that you're using, if you know that, and if you know the spectral profile of your autofluorescence. Okay, so now let's say you're using a tissue in which there is autofluorescence, and you're using 2 dyes, and let's assume these 2 dyes also overlap a little, and the autofluorescence is in the background, which is present across a wide range of wavelengths. It is anyway overlapping, okay, with both the dyes. But now if you know the spectral profile of your autofluorescence, and you know the spectral which you can determine, and if you know the spectral profile of your 2 individual dyes, and now if you take one picture, okay, in which you have all the 3 colors present: autofluorescence, then the signal from the first dye and the second dye, and now you run an algorithm. The algorithm, it determines what is in all those, the algorithm, it looks at each point in the image, and it identifies using a profile searching algorithm method. In this particular point, in one particular point, do I have autofluorescence there by its profile, or do I have or do I have the signal from the first dye or the second dye? If it detects that there is autofluorescence there, it subtracts it from that pixel. So the algorithm, it knows the total profile of the autofluorescence and looks at the entire image, and it looks at it point by point, and at each point, if it finds that there is if it finds, oh yes, I find that because there's a library, it knows the autofluorescence profile, and at each point if it finds that profile, it subtracts that light from that point. That's another approach.
Yes, sir, got it.
Yeah. So thank you so much, Amit, for a lovely lecture. I'm sure the children got a much better clarity on fluorescent cell sorting, and certainly you were very lucid in your presentation. Gradually you took them through. Certainly I'm sure they would definitely, and I would appreciate if you make a notes on what he spoke today, and if you send, Amit will be happy to go through it and do some correction if you have understood something not correctly. Is that okay?
Yeah.
Yes, sir.
Okay. So thank you so much.
Yeah. Okay.
Yeah. So have a nice day and take care of your health, Amit.
Yeah. Okay. Yeah. Thank you. So tomorrow we meet again at 10 and for the second part of this talk.
Yes. Class. And